Hello and good evening to everyone. Thank you for joining me. It could be anywhere in the world, so it could be good morning as well. Today I have two special guests, and they're special to me because they are willing to come and talk at very short notice. And so we have here two researchers who have been focused on COVID-19 since very early in the pandemic. And their interest and their determination to understand the spike protein is remarkable. And so within that framework, I'm going to be trying to take some of these complex subjects and see if I can simplify the ideas so that you can understand a bit more about if and what health risks could exist with regards to the spike protein. Now, you'll notice that I just said the spike protein. I have made no differentiation, and that's part of the discussion that we'll certainly be having with them. And so let's get them first on. We have Joachim and we have Mannan with us. And critically, I guess the starting point is to get them to introduce themselves so that we can get a better idea. Do you want to start, Joachim, and then get Mannan to do it afterwards?
Yes, thank you, Philip. Yeah, welcome everybody. My name is Joachim Gerlach, and I'm head of research and development at Vedicinals and collaborate with many different groups, medical sciences groups or molecular biologists and pharmacologists globally. And my goal, especially personal goal, was from day one of the pandemic, really like 25th of January 2020, to find a solution to combat this disease and everything connected with it. So what we are doing at the moment is we are still furthering the research and collaborating with many laboratories and universities globally in trying to really advance the knowledge of what is going on, what damages are occurring, and how can they be mitigated.
Excellent, excellent. And Mannan, yourself?
Yeah, I'm Abdul Mannan Baig. I'm a group lead of a infectious disease research group at Al Khan University. I belong from the country Pakistan, the city Karachi. I've been on to the COVID, long COVID, and then even, ME, CSF. Okay, these diseases are under my consideration now. I publish a lot on them, especially on long COVID. Okay, we have published more than 34 papers, if I'm not wrong. Okay, with two of them recently published on immuno-COVID, a paradox of autoimmunity and immune deficiency. So I'll be talking about the spike protein today. Your questions are welcome, and thanks for joining and listening to us. Thank you very much.
Excellent, excellent. So thank you very much, gentlemen. And what I will do is that we'll probably start off, Joachim, with you giving a kind of basic overview with regards to the spike protein. And just so people are aware, I'm going to try and see if I can break down some of the complexity. So I've got here a picture of the virus. So you see the virus, and it has multiple spike proteins on the top, and then a blown up image of what one of the spike proteins look like. And this here is the furin cleavage site. And the critical thing about it is that each one of these spikes at the top can bind to ACE2 and then enter the cell. So this is just one virus, and there are thousands or millions of viral particles that are produced in an infection. So that's the basic point with regards to this start. So let's bring in Joachim here and get him to tell us a little bit more about some of the effects that the research is suggesting can occur.
Yeah, so in our line of work, we have to, of course, look at all angles of this disease, be it the viral infection or be it just like the spike protein alone, free-floating, and or also through mRNA vaccines, which is also forming a lot of spike protein that are obviously also going through the whole organism. And so our first task, of course, was right from the beginning to look at the viral entry mechanisms and how can we block those, how can we block the binding of the spike protein, the cell membrane fusions, the replication of the virus. And lately also we are looking at what is happening with the vaccine lipid nanoparticle entry mechanisms. How does that work? What happens when the mRNA is being transcribed in the cell, and the spike proteins, how are they excreted? What is the exocytosis process in extracellular vesicles? exosomes and or other means. It's not only free spike protein. And the resulting conditions out of all this exposure to spike protein and or SARS-CoV-2 virus range from immunodeficiency, which again pulls behind opportunistic infections or activation of retroviruses like Epstein-Barr, herpes, shingles, etc. It goes into the epigenetic dysregulation, and also caused by the immunodeficiency plus some oncogenic mechanisms and deletions of tumor suppressor genes. We see a strong uptick in cancer being reported, already roughly 10% more cancer cases than before the pandemic. We are looking at all the thrombosis kind of conditions that are happening because of endotheliitis, endothelial inflammation, spike protein persistence in the endothelium, and that there is a constant flow of reports of microthrombi in long COVID and vaccine injured person. And there's almost nobody that is not affected. It's difficult to become later to that, how to diagnose the diagnostic tools for that. Then we look at neuro-COVID, because brain fog is not only like that you are feeling unwell like a hangover. There are really severe neurological damages to the brain, and in brain inflammation, microglia inflammation behind that. So we are very, very focused on that. Then we also check on all the other organ damages and see what can be done about that. There is a myocardial, cardiovascular, central nervous system affected, intestines, other organs, liver, kidneys, you just name it. So there are many, many organ damages already ongoing. And then, of course, we have the ongoing hypersensitivity, autoimmune reactions, and the hyperinflammation, and maybe some other smaller parts that we are the only group really looking into, is like the bacteriophage behavior and our bacteria being directly infected by SARS-CoV-2, again, contributing to the resulting immunodeficiency in the population, which is a big problem, as we can observe since quite a while. Yeah, so that is in a nutshell.
Yeah, so that is huge issues, you know. And
if you can bring that image that you were showing.
Yes. So before we come into that, I want to just before we start going into the details of the science of it, I just wanted to bring up this very important discussion here that you had mentioned, Joachim. And if you're not aware, Joachim is in Germany. Mannan is Pakistan. And you were saying that the health minister was speaking about this recently with some of the issues. I had done a presentation about what he said with immunodeficiency, which he subsequently withdrew. But what was he saying here, Joachim?
Yeah, it is now, of course, being reported that there are vaccine injuries, and also very severe vaccine injuries, mostly very strong neurodegenerative reactions to the vaccination. And so on German mainstream television at 8 o'clock at night during a big news broadcast, the German health minister spoke about that and mentioned that, yes, admitted that there are vaccine injuries, although he said it's not very frequent, but he's at least admitting that this is happening. And so they showed several cases on camera. They were describing their conditions, that they were permanently disabled, and so previously very healthy people. And plus he was like suggesting that the vaccine companies should at least donate money for research and to find solutions for the treatment of these vaccine injuries. So this is now being officially, let's say, confirmed by the German health minister and many other organs within the mainstream national media. Yes.
And so before we go into any of the details, Mannan, from the Pakistan side, you know, I mean, we're seeing quite a lot of issues in Europe and the Western world. Are you seeing a similar kind of pattern in Pakistan, or is it different?
I can talk about the continent, okay? In Southeast Asia, India is now again seen to be affected. There are lots of long COVID patients there, but in Pakistan, we don't have that many long COVID patients, though we have got COVID positivity going on. And since, like other first world countries, we have stopped checking the daily infection rate. Okay, so we don't have a national data emerging like we had in 2020 and 2021. But then I'll tell you something, things go sometimes subclinical. When I say subclinical, means not alarming, but I see people after the COVID infection getting lethargic, fatigued, okay, vision problems, taste problems, Even breathlessness is now persisting in many patients. So this is an issue which is not a continent specific one, but then specifically depending upon genetic makeup of a person, some of them can go into long COVID. Okay, and the incidence is very high in Europe and US and Germany and Italy.
Okay, right. So let's just get to what you were talking about with regards to your image here, Mannan. Let me just add this to this stream here. So
But
this is a complex image, and I just want you to try and see if you can break it down for me into some simple component parts. This here is the virus. These
yeah,
are the spike proteins, okay?
mhm,
Similar to what we've shown before. This
yeah,
is the ACE2
yeah,
and I guess NRP1, how it
mhm,
can enter into the cell, okay?
yeah, yeah.
Then you have a list of proteins across the top here. What are these?
Again, for these proteins, okay, Philip, if you use the word receptor, it is much better because they are expressed on the surface and they are normally responsible for the transport of many neuroendocrine substances and ligands into ligands means chemicals, agonist or antagonist inside the cell. So nature never made them for SARS-CoV-2. This virus is smart enough to not only use ACE2, NRP1, CD145, AXL, LFA1, LFA receptor, and you can see the list of them on the left hand side, and also some of them on the right hand side as GP41, ADAM17, like these receptors. Now, this is a figure which is 6 month old. I mean, if I get a cell membrane extending to the right corner of the screen and on the left corner of the screen, I would be adding 12 to 17 more receptors now as the data is emerging. First of all, I want the audience to know that this spike protein that they are showing here in red color and the one that you showed previously where you zoomed it out is made up of 1,273 amino acids. Now, these amino acids make a perfect coiling and turning and tossing of it, okay, to make it enable it to go inside the cell. So the thing is this, that be it a spike protein mounted over the SARS-CoV-2 or be it a spike protein that is resulting from the excessive synthesis of mRNA vaccine. And when I'm saying excessive synthesis of mRNA vaccine, try to understand what actually was the original claim. The original claim was when we inject it, it would be produced by the local cells in the deltoid muscle for a week or something or a few days, and then the immune system will recognize them. Once it recognizes them, then it's going to prevent any future infection and future infections by various variants because they thought that there would be very less mutation in the spike protein. Now, the thing which is alarming is this, and to some address they tried to address it, that the mRNA vaccine when made, okay, it was synthesized or configured in a way that it has got two protein substitution in the resultant S protein. So, which means in layman term, if you give the vaccine, the mRNA goes, makes the S protein, but that S protein has got two protein substitutions. Those two protein substitution, I should admit, were very smartly made in a region which actually disables the spike protein given or synthesized by the vaccine, okay, to bind to the ACE2 receptor.
Wait, I want
Now,
you to pause there a minute, Manana, because that's an
like,
important
so,
point. So you're saying here, I've just got the spike protein back again.
mhm,
This spike protein, when it was being, I guess, manufactured for to be produced in the body after
yeah,
vaccination, they
yeah.
made two adjustments on the top so that it would not bind to ACE2. Because my understanding, I've just got here an image of ACE2. The
Mhm.
ACE2 should normally bind to the spike protein, but you're saying that they made some adjustments so that this would not bind. Was this just the Moderna, or was it also Pfizer and the other vaccine manufacturers, AstraZeneca? Is that what we know?
Yeah, exactly. Be it Moderna, be it BioNTech, be it Pfizer, this two protein substitution had been there. I don't know why it was not so very commonly known because I find my peers and other presentation guys to go like receive this news with shock that okay, so it doesn't bind to the ACE2 receptor. It sits over the ACE2 receptor, but then it cannot go into that configurational change that enables the S1 and S2 to detach, and then the virus to enter inside the cell. Brings us to a question. And if I don't ask this question, somebody will, and if I answer it now, I possibly would be saving myself at least one question, okay, if not 99 of them, which actually everybody will ask. Big question: if you're making a mRNA vaccine to be injected here, which is just to programmed to produce a S protein for 5-7 days, which the immune cells should recognize and then kill the future prevent the future infection by killing the SARS-CoV-2 when you acquire it by infection. Why make this substitution? Because it's not producing virus. It's only producing S protein, isn't it?
Mm-hmm.
So
So no, wait, let's break that down again. So let's go again. So you're saying, why did they make the whole spike protein or
no,
just
why?
that?
Why make the two protein substitution or substitution in the furin cleavage site? Why make it? Because at the end of the day, the RNA given to a human as a vaccine is not going to produce a virus. It is going to produce only a protein known as S protein. That
Mm,
means
and mhm.
they knew that this protein can enter inside the cell. And ask me why I'm giving this much big smile, because this smile will in 5 minutes explain why we are having vax associated adverse effects in guys who are continuously producing that S protein in the body.
Okay.
So so
So
if
before you go any further with that, I'm going to bring up another image, and I'll ask Judge Joachim here. Just this is my work looking at when I was doing my presentations, and I've got here ACE2, which you said no longer binds because they've done the proline substitutions. Furin, you said, would normally bind to the furin cleavage site, and again they made substitutions here, so this shouldn't bind.
in
I've
some
also got,
in some latest vaccines, after the initial batch, they made this substitution. One of the BioNTech and Pfizer vaccine now has got some alteration at the furin cleavage site as well.
so that's a later present. So in the earlier versions, it was able to bind to furin, and there is neuropilin 1, there's DC sign, and you're saying there are about 8 other proteins, natural proteins, that will bind at various locations on
And
the
I'm not,
spike protein.
and I'm not saying it okay based on my educated guess or scientific guess. Each and every protein that you saw in that previous figure has got a PubMed publication over it. I mean, like, it's not my educated guess or my scientific predictions. NRP1, CD147, AXL, LFA1, all of these, I didn't choose them by some cherry picking. All of them have got at least 2 papers, or some of them have got more than 17 papers on them showing their role in binding the S protein and transporting the virus inside the cell. So,
Yeah.
I mean,
Yeah.
it's an educated guess.
When you heard about this, Joachim, what were your thoughts?
Yeah, it's of course you see all the experts were just concerned about the variants forming new mutations and new variants in order to evade the antibody reaction of our organism, and that is true to some degree. But the virus also is evolving now, producing new binding affinity to different host cell receptors, and so that also explains why you have different symptoms between these variants, because if you look at it, I think it's more or less 24 ACE2 independent host cell receptors plus some other endocytosis and or fusion enzymes and cleavage enzymes that play a role and can help the spike protein and or the virus to fuse with the cells. So according to the variant, it will then, of course, connect with these receptors, and these receptors are then differently expressed. For example, when Omicron came out, we saw that there were various proteins new on that Omicron that were not in any other variant. And I was discussing with Shankara, and I told him, I said, look, it's going to be more prone to destroy the blood-brain barrier, and it will be also more neurotropic because of the receptors that I checked where they are, on which organ tissue are these new receptors displayed. And so
Yes.
then you will have the damage there. That's why you see a shift in biomarkers, a shift in long COVID conditions and acute COVID conditions, according to the variant. That's explaining to a part why that is happening. So, you know me, we came always directly from the point of looking what kind of inhibitors can we provide with the protocols that we suggest for the doctors or produce ourselves even, in order to block also these receptors and these fusion and binding mechanisms that these newer variants are displaying, and also including the spike protein, the synthetic spike protein, because it is behaving in that regard like the virus. So if you're looking at bivalent boosters right now, they will have more possibilities to fuse to other receptors than the original vaccine from the Wuhan wild type spike.
Yeah. So here's a question that I want to ask, because I'm looking at some of the comments, and people are upset that they didn't know these things. But when we were looking at developing the vaccine, we're in the middle of a pandemic. We needed to have something quickly. Mannan, can you understand that there are multiple things that we could not, what would we would call the unknown unknowns? Is that a reasonable position to say? What would you say to that if the developers were saying, listen, there's only so much we could know for a novel virus?
For the virus and the protein both. Okay, I'll tell you something. Because when something is new, everybody is after to understand it. Okay, more do research. Okay, genome sequencing and understanding of this protein. I'll tell you something honest. Okay, and I don't know the reason for that. Is that we were not proactive. I mean, science has accelerated at tremendous speed, but the first 8 months after the pandemic actually broke out, there was a ear-breaking silence. I mean, like, I'll tell you something, and this after that, and then came the vaccine era. I don't know why it was not very candidly explained by peers and scientists from Pfizer and other companies. They could have taken a half an hour discussion session to educate the people what this vaccine is, why we made substitutions, what is our intention. Nothing like that. I mean, like you will see a 2-hour WWE wrestling match, but not a 30-minute educational, what you call as program, okay, in which the public would have been educated. Now, when you see surprises like these, that it is using more than 17 receptors, don't blame the virus. The virus is a space-occupying lesion. You know what I mean? I mean, we use the term in brain tumors, but then here the space is the Earth, and every species wants to occupy the space, okay, by even beating species like us, which we claim as the most encephalized mammal on the planet. I'm very sorry to say. I don't know which part of us encephalized and we evolutionized, okay, in that part. We are fighting this virus since 3 years, okay, without a single antiviral being manufactured by the big giant companies, okay, that we know. We don't know. I mean, like making 2 or 3 substitution, preventing it entering into the body. Why was a protein thought to enter inside a cell? I mean, why make those 2 protein substitution if you are not afraid that the protein would enter inside the cell and damage? I mean, like, I want everybody to ask this question: why
Yeah.
make substitutions?
There's a question that somebody asked, and I think that I have to. I'm trying to find it. It disappeared as quickly as it came. But here is the point: because we're talking a lot about what happened in Wuhan, this was the question: man-made virus. Have we ever seen this kind of pattern with any other virus where it uses this many entry mechanisms? Because I think even MERS only used DPP4. The original SARS CoV, I think, only used ACE2, unless I'm wrong. How is this one so evolved to have potentially multiple different routes of being able to enter the cells? Does that make any sense?
Do you really want to answer that question without getting us in trouble?
Well, I guess yes. I mean, I don't want yes, I don't want you to get us in trouble, Joachim. But I guess the question is, has that ever been seen before? I think that's all that we need to answer. That's what will make it. I have never come across this sophisticated kind of virus, but I don't study virology. That's where the question comes.
Nobody knows what really. I mean, there is documentation about the gain of function work, and it is extensively documented and visible for anybody that is interested. And you will find a lot of the features of the virus that we see in circulation today. You find those being experimented upon in these years between 2000 and 2019. Now, so there is no doubt, especially the furin cleavage site, and there are some other, let's say, worrisome inserts in the in this virus that, of course, are looking very odd, to say the least. Now, so I don't want to say that anybody has deliberately done anything here, but the chances of that has happened in the lab, and if you look at the sequences and what we see today out here as the virus, is too similar to be coincidence, to say something. Okay, this will be a debate for the next years to come, but look, we don't care. We look at the problem and try to find a solution, and we are looking at all these mechanisms, entry mechanisms, and we are blocking them so far pretty successfully. So we'll have another clinical trial coming up right now. I just signed the synopsis today, and we are enrolling now again acute patients, COVID patients, in another smaller trial, only 36 patients, but we want to also look at the bacteriophage behavior and the bacteria being infected, and how well do these different interventions work in stopping the viral reproduction and to stop the propagation of the virus. The virus has mutated lately, which is probably a result of the pharmaceutical 3CL inhibitors. So for the very first time now, I found papers in the last days that the 3CL main protease, which was unchanged pretty much through all coronaviruses, now has mutated, which is a big disaster, because now these drugs out there will be not working so well anymore. So we are constantly challenged, and we constantly have to update. But to come back, I don't know that there's any virus as sophisticated. Yes, there are other viruses also being that sophisticated. And if you look at the companions right now being triggered by SARS CoV-2 and or spike protein, like, for example, Epstein-Barr, also is highly carcinogenic, neurotropic, and does a lot of bad things, as bad as SARS CoV-2. So yes, there are other viruses that are even worse than SARS CoV-2 and more dangerous, and let's hope we will not see those in the next near future.
Yes,
Philip?
that's a good point. Go ahead, Mannan.
See, I would like to pick up from the line that you ended, okay? That SARS-CoV-1, okay, after that SARS-CoV-2, okay? Did we, I have a question, okay? Did we invest enough money, okay, or research and funds to study even SARS-CoV-1? No. The
Yes,
thing
yes.
was over. The thing was over. We left it there, we just left it there. That virus also has got a history of neurotropism affecting the brain. We had no progression of the study to observe its effects on immunity and stuff. The reason now we are being bitten by SARS-CoV-2. The problem is that the receptors that it is using and trying to use them to enter inside the cell, it will keep on changing. The virus is evolving. Okay, it will learn new ways to get inside the cell, including I don't know, even the cell membrane fusions. Okay, that won't need any other receptor at the first place. So now we have to just see. I mean, if you are not at the ahead of the curve, okay, you just sit and watch the show. This virus is very good to show you what it can do. I mean, like,
Yeah.
if no funding is there for drug research, synthetic molecules, herbals and supplements have been the guys who promote it, okay, and the ones who actually want to see their effects, none of them are receiving any funding.
Yeah.
I've been
I
shouting on.
think I think the point that has come out of the pandemic. I remember when 2 years ago, a group of us, we called it the Plan B group. We'd asked a very simple question, which is simply, what happens if the vaccine was not able to stop the pandemic, because we noticed that all the eggs were in the vaccine basket. And this was a very simple question at the time, and I'll probably try and see if I can get them back to ask this question again. But you're right, we almost, at the exclusion of the majority of other techniques, and I wouldn't even say just vaccines, I would say injectable vaccines, because I was calling for mucosal vaccines as another option that was not given the same amount of attention and focus. And it seems to have caught us out. Is that a reasonable perspective, Joachim?
I mean, if you first of all, the way this vaccine is administered is going to be very difficult to stop with an injected vaccine and create a mucosal immunity. That is not as easy. So your suggestion to go for a mucosal vaccine would be the king's way, because any other solution to that is secondary. Even what we are doing, we are treating the people that got infected. Maybe we can prime them a bit before they get infected so that they can handle the COVID infection better. But we cannot avoid infection. That can only be done with some nasal sprays or very excellent masks and some other measures that could help to bring down the infection. But if you look at, it's very simple at the moment. You don't have to be a scientist. If you want to know what's going on, for example, you look at the loss of working hours in 2022, for example, or you look at the amount of infectious disease hospitalizations apart from COVID, skyrocketing. If you look at the new reported, or let's say reported cancer cases in the US, you look at the data from the American Cancer Association, you will find that from 2021 on, you have a 10% increase annually, now stable 10% over the usual average in reported cancer cases. And so the list goes on and on. So if you pay attention and you look at just the normal available data, even excess deaths, and whatever else is reported, then you know that this is causing a lot of, let's say, widespread damage, be it the infection and or the spike protein. I don't know how much, nobody knows really the real numbers about vaccine injuries. It can be from a very small number, ranging to a higher percentage, even as long COVID. We don't know. That will come out in time, and then it will be shown. But the bigger problem that we can see at the moment is also that that there is no herd immunity in that sense. So, because if we would have herd immunity, again, you don't need to be a scientist. You just go to the Worldometer, COVID cases being reported, and you look at 2020 and you look at 2021. There is already quite a lot of cases, yes, millions. And then you look at 2022. So if there would be any immunity through vaccination and or previous infection, we should see a drop in the overall cases in the population. But we see actually in order of magnitude more COVID infections in 2022 than ever before during the pandemic. So obviously there is no immunity, otherwise this would not happen. It's very simple.
Yeah,
I don't need to be
I'm going to bring up a picture here, and I want to ask you about the impact on the immune system, because this is what the German health minister had said as well. And you see here, this is the bone marrow, and this is the stem cell. It breaks into two groups, the lymphoid and the myeloid, and then it goes down to produce neutrophils, monocytes, basophils, and on the other side, the T cells, the B cells, the natural killer cells, and the plasma cells. So if we look at this whole picture here, which immune cells are primarily affected, or is it all of them? Any thoughts on that, Mannan?
You pick up the macrophage and the dendritic cell from the left-hand side, okay? And you pick T, B, and NK cells on the right-hand side. There has been a disproportionate ratio of CD4 to CD5 and CD3 and CD8 cells that have been reported in long COVID patients and patients who are producing excess of the S protein after vaccines. Macrophages are studied with the spike protein, be it infectious or be it the excess production of the S protein following the vaccine. And these cells, okay, I want to bring something very important here, okay, Philip, if you can, and your audience can understand that. And please feel free to ask if you didn't understand. I will go into details of explanation. There are two types of deficiencies of these cells. Number one, virtual. Virtual means their count goes down,
Yeah. Hmm.
lymphocytopenia,
Yeah.
okay, you call it, or neutropenia, or
Hmm.
you call it eosinopenia. There is one condition, okay, which we have mentioned in a recently published paper, okay, on lymphocytopenia, that these guys are suffering a condition known as functional lymphocytopenia. Million
So
dollar
that
question.
means, so these lymphocytes here,
Maybe there
although they are there in normal numbers, they are not functioning
function.
effectively. So just to make it clear for everybody, these T cells target the virally infected cells and kill them. The B cells here will be making antibodies.
Antibodies.
And these natural killer cells are some very specialized all-purpose lymphocytes. And so this is the backbone of your very sophisticated acquired immunity. The cells on this side tend to be more general purpose immunity, but in terms of very specific immunity, it tends to be these lymphoid cells. So you are
Yeah.
saying that these cells, even when the numbers are normal, are not effectively functional.
Yeah, and the reason to support that is that many peoples with normal T and B cell count are developing reactivation of the mono, that is EBV. They are developing reactivation of HSV. They are having Zoster's back. I mean, what happened? I mean, like the immune cells are there, so there is a dual factor going on: quantitative lymphocytopenia, or I call it quantitative immune deficiency, and qualitative immune deficiency. And I'll tell you something: qualitative immune deficiency you cannot diagnose. I mean, like, you rely on these cell counts. There is only one way, that you take out these T cells or B lymphocytes from the body of the person, put them in a petri dish, and challenge it antigenically with any virus, including SARS CoV-2. If they fail to respond, that's the functional deficiency. If you put it back in the body, you will be counting them, that yes, it is there, but a microscope cannot tell you that it is qualitatively damaged. Qualitative damage should be checked. In that paper that we published, we have suggested very strongly that these guys who are having normal count of lymphocytes in their body, the lymphocytes should be brought up back into the petri dish, challenged antigenically, and if they don't react, that means these are the qualitatively injured guys. I mean, like with lymphocytopenia and immune deficiency.
So
Horrible.
someone has asked this question here. What would you say to that? There is a strep outbreak at the moment. Is this at all related to what you're talking about?
See, the medical books taught me one thing, in 24 hours, if bacterial infection is not combated well by the neutrophil, after 24 hours, the macrophage will come. And did you see in the previous diagram, my cell of choice from the left panel was macrophage. Macrophage are also qualitatively and quantitatively damaged. If the macrophage cannot come and fight the fight of the neutrophil along with it, you just can count of a 6-month prolonged strep infection as well. So no surprise if a bacterial infection is not getting eradicated. And never to forget the fungus that you saw in India in 2020 and 2021, that black fungus, or whatever they call it, mucormycosis. It's on the kingdom of its rise. It's claiming its patient. Why people are having oral and candidial infections and all of these with these fungi? These fungi, we kick them off in 12 hours if we have a intact immunity, qualitative and quantitative.
Joachim, you're going to say something here.
Yeah,
Please.
because that is actually really an interesting angle on this whole and completely overlooked. And you were hosting us when in the conference where Carlo Brogna was presenting his findings. So if the bacteria in our gut plus in our respiratory system, the biome is affected by the virus directly through infection of the bacteria, and so they are a reservoir for the virus, same as our host cells. So I have collected most of the data available on post-viral or long COVID biome. And it shows very clearly that you have a very strong dysbiosis resulting, and not only from the inflammation, but also through the direct infection of this bacteria by SARS CoV-2. And of course, it is not a difficult thing to imagine if you have now all the beneficial bacteria, butyrate producing bacteria being massacred, you have a high amount of toxic peptides floating around in your guts. And of course, then you have an overgrowth of bacteria that is pathogenic, like strep A. So if you ask my opinion, it's a mix of the immunodeficiency by direct infection and invasion into the T lymphocytes, which you can see again under the electron microscope. So both things I mentioned, I have plenty of slides, electron microscope, T lymphocytes being full of SARS-CoV-2 virins. You can see macrophages being directly infected, full with virins. You see syncytia formation, cell conglomerates of dysfunctional T lymphocytes by the spike protein and or the virus. You see bacteria full with SARS-CoV-2 virins, breeding them. You can breed them in a lab, in a petri dish. You can breed the bacteria and the virus at the same time. So how that cannot affect the immune system, wouldn't have any answer. Of course,
this is a beast of an infection, and in effect, as I look objectively at what is happening across the world, the most important piece of the puzzle seems to be that your mucosal immunity has to prevent the virus from getting into the bloodstream. That seems to be so. In countries that have herd immunity, naturally acquired herd immunity, I recently spoke about Papua New Guinea. I think they have technically the pandemic is over because the virus can no longer spread, and then the virus technically becomes ineffective. What do we, however, do in situations where that mucosal immunity is no longer as efficient against Omicron? Because this is what we're seeing in the Western world.
May I say again a word? Because most people that now know about Carlo's finding, they just concentrate on the intestinal biome. But of course, we do have a very sophisticated and very similar biome in our nasal cavity, in the olfactory system, and in the respiratory system. So if these bacteria are getting infected, as they do in the gut, then of course you will have a less mucosal immunity, because the biome in our respiratory system is also contributing strongly to our immunity. Plus, people don't understand also SARS-CoV-2 is not only damaging immune cells and the biome, it is also suppressing type 1 interferon very strongly. It's a smart virus. So as soon as it enters, immediately it is kind of neutralizing your type 1 interferon responses.
Yeah. And
it is an ongoing process afterwards. So it is hitting every button that is possible to make you susceptible for repeated infections.
Really right.
Sorry, Mannan.
Also, the cell that you showed, B lymphocyte, on that diagram. Can you just fetch that diagram, please?
Yeah.
That B lymphocyte that you see here is responsible for production and normal maintenance of an antibody called as IgA, the mucosal immunity antibody. So just imagine that if these cells are sick in the mucosa and they are normally responsible for the production of immunoglobulin A, and no more immunoglobulin A is there, what will happen to the natural immune or mucosal immunity of the person? It would be down. And then you can count on the number of the bacteria: streptococci, staphylococci, meningococci, gonococci, you name it. Okay, because the first hand we deal with them with IgA, they do not progress to infection. So this is a immunocompromised state now. I mean, like, if you don't do anything up for this, this is you will not call it immune deficiency AIDS-like thing, but then tell me, how is it anyway different from that?
Yeah. So listen. I mean, we already almost coming up to the hour mark, and one of the areas that I wanted to touch on was a question that has been floating around for some time with regards to fibrin clots. Now, I've seen where there have been talk in terms of in mortuaries that the clots that they're pulling out are quite thick and very difficult to break down. Is this at all related to anything to do with the spike protein?
Everything related to the spike protein, not anything, you know. And why I say everything to do with it, I will put it simple, okay? I'll not put a complicated description here. In our body, okay, anything, bacterial toxin or an antigen antibody reaction, anything which damages the endothelium automatically starts the cascade of thrombosis. Okay, first of all, I would like to use it, the term thrombosis, only because clotting is outside the body. When you actually just prick your finger and a drop drops down and it becomes hard inside the body, if the same mechanism makes it, if it makes it, okay, we call it thrombi. Now, you know, if you just like say thrombosis is the cause, I understand, and those clots should be either called abnormal clots or they should be called as thrombi. Now, thrombosis is a consequence, it's not actually the effect. The effect is the damage to the endothelium. $1 million question: what is causing the damage to the endothelium? The S protein from the infection and the overproduced S protein from the vaccine. So
And
yeah,
I just want people to understand when you say the endothelium, I've got here a picture, a cut through section of a blood vessel. So this
Mhm.
is the muscle on the outside. And on the inside here are these endothelial cells.
Very
And
right.
this inside is usually very smooth, like glass. And you're saying that anything that damages these cells will
Mhm.
increase the risk of clots being formed.
Yeah, increase the risk means in 1% they would not be formed. So it doesn't increases the risk, makes it absolute that the thrombus will be formed.
I
Because
see what you mean. Yes.
yeah,
So it
you know,
has to occur. Yes.
it has to occur because nature made it like that. When you prick your finger, okay, and you damage the endothelium, and the drops down on the glass slide, the platelet come here, okay, and fibrin come here and close that defect. Okay, this is normal. We call it
Mm.
clotting. You know, when the same mechanism starts appearing in your body without a damage to the blood vessel in form of it is causing bleeding, you call it thrombosis. And spike protein is an enemy of endothelium. Just imagine, okay, I'll very quickly give you the sequence, okay. Spike protein in the blood, one, uptaken by endothelium, NRP1 receptors, CD147, whatever receptor uptakes, enters inside the cell, proliferates, apoptosis, endothelium ruptured. When I say endothelium ruptured, basement membrane exposed, thrombus will form there. You like it or not, okay, it's the consequence of it. Let's go to the spike protein by vaccine. Spike protein from vaccine being overproduced, S protein in the blood builds up, it damages the endothelium by directly interacting with the cell membrane, uptaken by it, or even causing irritant effect on it. Thrombosis follow, and the blood vessel is closed. And you can ask me a question. Can you clinically prove that your statement is correct? See what is common between COVID and the vaccine injured guys. Thrombosis, heart attack, stroke. All these thrombi in the brain, stroke. In the heart attack. Okay, in the leg, gangrene. In the intestine, bowel ischemia. So, I mean, like the consequences are similar because the common denominator is the S protein. You know what I mean? So, with virus, the damage is more, but with S protein, the damage is not less. Because if you kill the endothelium, every other organ of your body is going to collapse. That we are observing at present.
And so one of the things that people always say when I'm looking at comments and so on is the perception is that this happens to everyone. And I say no, it doesn't happen to everyone. There's usually a percentage of the population that is more at risk for adverse outcomes. I think what we don't understand is whether or not that percentage is 0.1, 1%, 10%, or even 30%.
Only because we didn't do any funding to support those studies. You
Well,
asked me this question 2 years back, Philip. I remember that day. I told you that if they don't invest in investigating the HLA predisposition of guys for COVID, long COVID, and vax injuries, we'll be sitting here after 2 years and asking the same question. See what's happening. We are again sitting here asking
and
the
we're
same,
asking the same question.
same
What are
question.
your thoughts, Joachim?
Now, look, it is, of course, also a question. Many of the conditions arising from spike protein and post-viral conditions, the diagnostic tools are not there yet, really. Now, for example, for the microthrombi, the only thing that works, what you see Receptors, Beate Jäger and Butrino and others use, is the fluorescent microscopy, because there you can really stain the particles that you find. It's platelets, or it's fibrin, or it is some other material. Some of the stuff that Beate Jäger, for example, I was at her lab many times now, and Mannan will join her now also to work with her. They don't know what it is really, but they find microscopic thrombi in pretty much all patients. And the worst thing she told me, like a few weeks ago, she called me explicitly for that, and she said, Joachim, I find endothelial debris in every blood sample I investigate under the microscope.
She will.
Yeah.
And so just so people understand that, when you say endothelial debris, you mean that the lining of the blood vessels that I showed previously is being damaged, and the pieces of these cells are being consistently found. And is this across the board or only in patients who are unwell?
Across the board. And she says, I hardly find any. She's desperately trying to find blood samples from uninfected people or people that don't show that.
And so this is even with just infection.
Yeah, sure.
Just infection as well. And so one of the difficulties, and so I'll ask you this here, because now we're going to, so I use my control group at the moment is Papua New Guinea. It's only 4.15% vaccinated. Are you telling me that they will have had Omicron all through the population by now? So if we looked at their blood samples where they have natural immunity, will we see the same pattern? Or is this because we are seeing a combination of the hybrid immunity?
Look, I don't know, because we also talk a lot to our colleagues in India and in Pakistan, and obviously they have a lot less problems than we do have in the developed world. That has different factors. Yeah, we know we've been investigating before COVID, and we always were screaming from the rooftops. We have around 50% of the adult population, or even the general population, already with chronic disease. 50% bloody already taking some medication for chronic disease. That is, of course, a different picture in Africa, in India, or in Papua New Guinea. So the population there in general is much healthier. They have a much broader biome to start with, even almost double the biodiversity in their biome compared to us Westerners. Plus the age factor, the demographics play a huge role. But I think that the underlying inflammation caused by industrial food and this bad lifestyle that we have, and the environmental and toxic load on our population, is actually the breeding ground for all these chronic things we see. Because if you have an inflammatory already underlying condition, and then you add neutrophil elastasis, and you start having spike protein in the endothelium, it will fall on fertile ground and cause much more damage.
Yeah. And actually, I tell you one important thought that I was reflecting on before we had the conversation is, if the spike protein just on its own is as lethal as we are almost thinking, then one of the groups that should be very significantly affected are neonates, because they have such a poor immunity, and we don't see that. So I think that what you have said, Joachim, when you have underlying inflammation and you have the inflammation associated from the spike protein, the combination of the two makes an explosion. Would that be about right, Mannan?
I would say 80% of the cases, yes. Okay. Why? Because if you are already compromised by comorbidities, by which I mean several other ongoing illnesses, you are a very susceptible prey for SARS CoV-2. But then you see, okay, very healthy individuals, okay, dying of sudden cardiac death. I mean, athletes, trained athletes, how do you explain that? I mean, those guys have got a very balanced diet. They refrain from processed food and stuff. They keep a check on their diet, okay, plenty of fruits and everything, and dropping dead. in the ground. So it try to understand that it's a combination of a viral load or the S protein load and underlying comorbidities which plays a role. And those,
Excellent. Mhm.
and those
But you,
combinations,
yeah,
yeah.
there was one more thing that we jumped over, and that was you were asking actually about the large thrombi and
Yes, yes,
the rubber-like
yes,
consistency,
the rubber like. Mhm.
calamari. So I spent the better part of a year just looking into that, half of the day more or less. So if you look at the microthrombi that are being reported and seen under this fluorescent microscope, that is probably one part of it. These are the building blocks that you can later then accumulate. But
And so, are you talking about this image here?
yeah, for example, that is a typical, that was a case report where we had a British UK doctor, long COVID sufferer, and he was having these microthrombi for a long time. As long as Beate was looking at the slides, they persisted, even with some therapy there, triple anticoag
And
therapy.
these are the microthrombi you're talking about?
Yes, exactly.
Mm.
These are the microthrombi, and so they are also consisting of material that is difficult to disintegrate for our own antithrombotic system in our body. And what we did on the lower slides, you see, we treated him with our product for 20 days, and that significantly improved, for example. So there is and so maybe we also were good at dampening the endothelial inflammation and helping this. There's like at least 50 different pathways leading to these different thrombi formation. Now we have published on that. It's still in peer review, but it'll be out very soon. Plus, then if you look at these microthrombi, and then you see what happens when you take the spike protein and the neutrophils, these immune cells that are being triggered and activated by the infection, by inflammation, and by the spike protein directly. Once they get triggered, they are releasing an enzyme. It's called neutrophil elastase. And when I saw the work of Dr. Hammerstrom and Sophie from Sweden, and the electron microscopy pictures of the fibers that are resulting from the combination of spike protein and the neutrophil elastase, it blew me off my socks.
And so just explain this picture for us here, Joachim. So what are these here? These are the elastase fibers?
No,
These are amyloid
no.
fibers.
It
These
is
are
actually,
amyloid fibers.
it was a spike protein. So it is kind of a shape-shifting process. You add neutrophil elastases to the spike protein, and these fibers will result, these branching fibers.
So you are saying that this was a spike protein?
Yes, sir, exactly.
So this was another spike protein?
Yes.
Many of them. So when the elastase is added to it, it becomes these insoluble fibers.
Exactly.
Amyloid fibers, amyloid fibrils. He calls
amyloid-like fibers,
yeah.
not amyloid.
Yeah, amyloid-like fibrils, or you can call them amyloidogenic type of fibrils, okay, which resemble the Alzheimer's related amyloid in the brain.
And so is this related to those very thick, undissolvable clots that they have been talking about?
$1 million question. That's why now there are several samples on the way to the electron microscopy to verify if they can find now these fibers in there. But I wanted to explain what my hypothesis is. So if you have ongoing already microthrombi and thrombi formation, and you are a person with a very high inflammatory levels in your body, which will of course mean that there's LPS involved, and that the neutrophils are in general already overactivated. And then you get a high load of spike proteins, and the neutrophils themselves have a FCY gamma receptor on top of them. And if the spike protein hits directly on the neutrophil, it will be triggered. Yeah, same like the mast cells, for example. That is also happening. So this neutrophil elastase plus the netosis, which are very fine webs that the neutrophils are excreting, this will become a mix of different fibers, of different compositions and platelets, and you just name it, endocellular debris, forming these clots.
But the reason you cannot dissolve them, because conventional thrombi, conventional clots are destroyed by plasmin, fibrinolytic agents like fibrinolysin, urokinase, streptokinase. You put a drop of it on this, okay, and this doesn't move a millimeter. Why? Because this amyloidogenic type of fibrils, they are very resistant to digestion by conventional fibrinolytic system.
Yeah.
Wow.
So the next step in the labs now is to see what can take these kind of fibrils apart. And we have several other also enzymes that we want to check on the spike protein itself. We have other components we want to check if they can clear the spike from inside the cell, from the cell membrane. Then you want to also, once you get spike out of the cells, you want to be sure that even if it's fragmented or in one piece, it can still then go to other cells and cause a lot of havoc and or autoimmune reactions. So we are looking at what kind of molecules can kind of wrap in these fragments or the whole spike and bring it safely out of the bloodstream into the urine or whatever excretion process, so that we really start now working on reversing the process and getting rid of these constant and persistent, let's say, proteins in our body. So it's various
Let's update Philip on this, that Dr. Beate is ready with these microthrombi in her lab with spike protein broken into these amyloid-like fibrils. I'm going to go and test them on Petri dishes to dissolve them by usage of several enzymes and nutraceuticals and stuff. And the moment we are successful, okay, we'll share it with you on this platform even before the publication. So just
that would be wonderful. So listen, guys, we're just over the hour mark. I want you guys to give us a closing as to where you think we need to be going in the near future. Who wants to go first?
Joachim first, please.
Yeah, I don't know where we need to be going. We are going the whole time. So we have not really stepped still in 3 years, not a single day. And we will not rest until we find out what is going on, until we find solutions. And the collaborative network is getting better qualitatively and also from the expansion that we have now right now. So it is also a lot of doctors like you, many like integrative medics. And let's put it that way. It is such a complex problem with hundreds and hundreds of pathways in dysregulation. I think that we should find what I call the common denominator protocol that will help a big part of the people suffering in order to buy capacity and time for then more specialized and more individualized medicine to be applied to the cases that are more difficult to treat. Because the sheer numbers of people that we are looking at with damages and ongoing health problems is in the probably way above 100 million globally, especially in the Western world. So where are all the doctors? Where are all the laboratories? Where are all the clinics that are qualified and have time and would be able to help these patients? They don't exist yet.
Yes, quite right. And so, Mannan, if you are still there, yes, come and give us your closing thoughts.
You kill the beast, okay? You don't kill the smoke, you know. So if you cannot extinguish the fire, don't complain about the smoke, you know. Finding microthrombi is very good. Dissolving them is a very excellent thing. But are these the cause? Are these the cause or the effects? The answer is it's the effects. So $1 million question, what is the cause? So if you know the provocating agent is SARS CoV-2, if you know the provocating agent is overproduced spike protein, and you find a way to absorb it and excrete it in the urine or disable it, okay, in some way, that actually is the key answer to the question, that will we win over it? We have to identify the persistence of the virus and the overproduced S protein as the cause, and then the treatment. It's not far. I mean, like, at least I could say that we have progressed since last 2 years, okay, when I was sitting here in one of your talks. So it's moving ahead. And
moving ahead, and sadly it's not as quick as people would hope, but we at least we're moving. And so I thank you guys for coming at this last minute, being able to share this research, give an insight into the challenges that are ahead. Really appreciate it. Would definitely look forward to hearing what the findings are with regards to dissolving some of these amyloid fibrils in the future. So we'll certainly look out for that space. So thank you everyone for being with us. And we will certainly be keeping you updated. And thank you, Abdul and Mannan and Joachim. And if you'll just hang on a minute, let me just do
Okay.
the outro for the audience. Have a great evening, everyone.
Thank you.
Thank you.